cells in the population. And there are a number of ways that we can measure the growth. A couple of those are the number of viable cells, the total number of cells, which would be the living and the dead cells, and we could also measure the biomass, or how much does the mass of the cells weigh. Bacteria are always going to be going through this system of binary fission. And so you'll see at the top you have a single bacteria that has one single chromosome. Next, the DNA will get replicated, giving us two circles of DNA. They're two copies of the same thing. And at the same time, the cell will elongate some. We'll next get a septum formed where a wall is being built between where the two cells will come from. We finally have a completed septum, giving us two completely different cells, and then the two cells will move apart, really seeing how independent they are. One of the things that you can notice in this picture is that of that green circle that represents the chromosome, there's a black dot, and that black dot is representing the origin of replication, or in other words, where the chromosome starts replicating itself from. And that's a real interesting scenario because we always think of just a circle as not really having a starting and ending location because it's a circle. The origin of replication, however, is a certain DNA sequence, and you can see that that origin of replication will always be affixed to the cell wall. And when we start forming the septum, we're going to move one copy of it to either side so that when the whole septum is formed, that we're going to get one chromosome in each of the two cells. One of the things that is real important in binary fission is that you'll be getting an increased number of individuals as the population will grow, okay, and that growth is logarithmic. So here, when something... Yeah?
Well, oh, I'm sorry. Just checking and I'm going to get your trash now, okay?
Okay. As you can see, and using the picture from before as the example of binary fission, every time you have a division, you increase the number of cells by twice. Each one cell will go to two cells will go to four cells will go to 8 cells. And you can do that up to a certain point where you get the carrying capacity of the media that you have. But basically, in just 10 hours, in this example, you'll get over 1 million cells produced from just one single cell. Okay. And that's one of the great joys of working with microbiology, is you can get a lot of starting material very quickly with just a few hours of growth. Here are the numbers at the very top, and at the bottom uh, we have a comparison of if you were to graph that data on a logarithmic scale or on an arithmetic scale. So when you look at the graph using the left number of cells, okay, that is the arithmetic scale. So 1,000, 500, 100, and you can see that that green line is an exponential curve. Okay, so as you were increasing the number of cells with time, you're getting exponential increase. Now, if we use the um, log scale, okay, so now you're looking at the graph on the or the scale on the right. Now you get a completely linear line if you're looking at the same culture with on a logarithmic scale. And we almost always use the log scale because it's just so much easier, and doing the math, for example, is so much easier on a logarithmic scale than if you're using an arithmetic scale. The bacterial growth curve is divided into four parts. First is the lag phase. The second is the exponential phase. The third is stationary phase. And the last phase is the death phase. And I think it's important to note that this scale is the log base 10 of the number of cells. Okay. So we go through first the lag phase where there's almost no growth, and mathematically the numbers just aren't there yet to see a big increase. Now we go through this exponential or log phase, and in the log phase these cells are increasing in a logarithmic pattern, and that should be a straight line when on log paper. Then in the stationary phase, the cell number remains constant. And this is a kind of exciting stage I'll say in just a minute. And then finally, we have the death phase. Now, in the stationary phase, it's important that that number of cells is constant and that it is a constant one cell grows for every cell that dies. So there's a somewhat of a turnover, but mostly the cells are just sitting there. Okay, for every cell that dies, one cell is born. Now when you get to the death phase, you're now increasing the deaths over the cells that are being made, and so you get a total decline in the population. Now, one of the reasons why we might wonder why we care about that, and I think one of the best examples is with food poisoning. So let's say that the cook is making some curry at 5 p.m. and that person didn't wash his or her hands, and so about 10 salmonella are introduced into that curry. Okay, and I in part chose a curry because it's a nice growth medium. Okay, so 2 hours after that, those 10 salmonella have now reached a population of 10 to the 4th cells per ml. And these this population size won't really affect many, if any, of the customers. At 9 p.m., the population has reached 10 to the 8th. And in that case, everyone who's going to eat at 9 p.m. is going to get sick. And this is one of the things when people are in the habit of eating, say, after a movie, uh, people tend to get ill after they when they eat after a movie, because if the food got contaminated early in the day, it'll get uh, the cells will allowed to grow. Hence, they can give a population size that's great enough to get everyone sick. So that actually brings up something, the 240-140 rule. And this 240-140 rule is a great rule and one of those things that you should really take home from this course. Okay, so the 240-140 rule is don't eat any meats, dressings, salads that have been kept for more than 2 hours, that's the 2, between 40 Fahrenheit and 140 Fahrenheit. Okay, so that means you can keep things in the refrigerator, which your refrigerator should hopefully only go as high as 40, and you'll be able to kill things if they're heated past 140. So as long as you don't let them grow, let the bacteria grow, okay, for more than 2 hours in that critical period, in that critical temperature range from 40 to 140 Fahrenheit, you should be okay. But if it does sit out for a couple of hours and you're just not sure, it's always best to just throw it out. The possible generation times for different kinds of bacteria is very, very different. For example, one bacteria, Clostridium difficile, will divide about once every 8 minutes. And one of the reasons why I bring that one up, not only is it one of the fastest things that we know how fast, it divides very fast, but Clostridium difficile is an incredibly dangerous intestinal parasite. It's about 90% fatal. I've actually am one of that 10% who survived that, but it was not fun getting through that. Okay, a very typical bacteria is E. coli, and it is going to divide once every 21 minutes. Now, since we already had a disease state, the Clostridium botulinum, that is the causative agent of botulism, and it's going to divide once every 35 minutes, and so not quite as fast as the previous examples, but it's not all that long. And then the extreme that I'll show you here is Mycobacterium tuberculosis. And Mycobacterium tuberculosis is the causative agent of the disease tuberculosis. And it only divides once every 121 minutes, or in other words, every 12 hours. And so one of the reasons why when you get tuberculosis that you have to take antibiotics for so long, and as we'll learn in the near future, antibiotics are only effective when the cells are actually dividing, and so if it only divides once every 12 hours, then you don't have that many opportunities where the cell is dividing so that it can kill the cells. Now, I had mentioned that we could measure with either direct measures or indirect measures, and here's an example of an indirect measure. So on the right side, you can see that you could have a light source. This is called a spectrophotometer. Okay, and you have a light source, and then your test tube with the objects that you want to know how many are there in it, and then something that's going to detect the light, and it's going to give you a readout by how much light is being able to pass through that sample. Okay, so on the top, on the right side, when you have very few cells in the test tube, the cell doesn't really, or the cells don't break up the light, and so a lot of it passes through, so you get a very high reading. On the bottom, where we have a lot more objects, now there are going to be more objects to block the light from getting to that detector, and you'll get a very low reading. By eye, it's harder to quantitate, but you can see those two panels. The leftmost being a very heavy solution of cells, where there are a lot of cells in that test tube, and the other, there are very, very few cells in that test tube. And what you can see is that when you have a lot of cells there, you can't even see through it, right? You can't see the lines behind the test tube. And so it's the same thing as what's happening in the spectrophotometer, that the light is being scattered in such a way that's not seeing the detector, in the same way that you can't see the back of the paper behind the test tubes.